Hello everybody, how is everybody doing today? This is Dr. Manoj Shah. We are going to talk about a topic that has been discussed mostly in lay journals and public with little evidence from medical science. So we are going to today talk about any scientific evidence about leaky gut. There's all this fuss about its role in maintaining health and causes of many diseases. Let's just talk about what we know so far and what can we do about it. Because we are talking about leaky gut, first we want to know what are the functions of the gut. Gut is gastrointestinal tract, and if we ask today, I know most of you will be talking about the importance of gastrointestinal tract in allowing us to take food by mouth, that is intake from oral cavity, then transport of the food from mouth through esophagus into the stomach and further down the tract. Then the most important function people think about is digestion. And there are two types of digestion. First is mechanical digestion, that is mainly in the stomach, and then chemical digestion, that is digestion of food by different enzymes, and these are the organs involved are small intestine, where the bile from the liver, the digestive enzyme from the pancreas, and intestinal epithelium has some enzymes, so they help in digesting the food, and then absorption of these nutrients from small intestine. And the last part is excretion of undigested food from large intestine or the colon. However, we don't many times think about it or not say the most important function of the gastrointestinal tract in my mind. This important function, I think, is of intestinal tract acting as a barrier. Intestinal tract is a barrier to the entry of any outside material to the inside of the host. This is very important function that host or our body does not have to deal with any external material that has not been selectively absorbed by the intestine. To understand the intestinal barrier function, we have to understand the anatomy or histology of the small intestine. If we look at small intestine, it's not just made just like a pipe, but there are multiple folds in the intestine. If we look at those folds, these are the folds in the wall of the intestine. If you just open up the intestine and look inside, it's not smooth. There are many folds on it. Even each fold has many folds on it. And if you look at these folds, even they have many folds on it. So there are many folds of intestine. This allows to increase the surface area of the intestine. When normally the intestine is about 22 feet long, small intestine is about 22 feet long, so large intestine is about 5 feet long. But if we stretch out the whole intestine with all folds removed, if we just kind of stretch it out, then the area of intestine will become almost like a half of badminton court. So it will be close to about 300 to 400 square feet. That allows a lot of area for absorption of the nutrients. Now, if we look at this intestinal these folds, if you look individually, one fold is called villus of intestine. Many folds together are there, we call them villi of intestine. But if we look at this fold, there folds have many cells on it, and some of these important cells of intestine, these cells of intestine, they also have folds on it, and they are called microvilli, and we will look at that later on. So the intestinal barrier function is secondary to the development of these specialized cells and the folds and villi and different things. So we'll kind of talk about it in next slide. So what is intestinal barrier? It is a functional entity separating the gut lumen from the inner host and consisting of chemical like mucus, biological like microbiota, mechanical epithelium of intestine, and immunologic elements which are lymphocytes and innate immune cells in the intestinal wall. The intestinal barrier is the main interface between the external environment and the host and maintains an equilibrated homeostasis by allowing the passage of selective nutrients such as amino acids, carbohydrates like glucose, and different type of sugars, electrolytes like sodium, potassium, chloride, calcium, magnesium, lipids like fats and water, while also hindering the entrance of toxins and bacteria. This intestinal barrier is made up of 3 layers if you want to think about it. This is the intestinal lining. The part towards the intestinal lumen or cavity is this part, and this part is intestinal wall attached to or communicating with systemic circulation or blood vessels. So first important kind of barrier function is maintained by biochemical barrier. Biochemical barrier, these biochemicals are in the intestinal cavity. They're made up of biologics or microorganisms that are in the intestinal lumen. There are some good bacteria, there are some not so good, and there are some bad bacteria. These bacteria alter the response of other bacteria also. So it can help in maintaining a barrier function. The other part of biochemical barrier is the mucus. Mucus layer in the intestine is made up of 2 parts. One is inner mucus layer, which is thick mucus layer, and outer mucus layer is thin mucus layer. Mucus is secreted by these yellow cells, we call it goblet cells in the intestinal lining. The second barrier is the most important physical barrier function of the intestine. This barrier is made up of intestinal cells lining the whole intestinal tract. There are many different cells. The most important in small intestine are called enterocytes. These are enterocytes. They have small folds on themselves, also called microvilli. Then there are intestinal M cells that are responsible for maintaining immunological function. There are goblet cells, there are lymphocytes, there are Paneth cells. All different cells are in the intestinal lining. Now all these cells are attached to each other by these little different lines here you see. These are called tight junctions. These are the junctions between the 2 cells, and there is no gap left between the 2 cells for anything to go across between these 2 cells. It is very important that this tight junction is maintained, so there is nothing going through the intestinal lining into the intestinal wall. If anything goes through it, then there is a third barrier. There is immunological barrier. These immunologic barriers is made up of many immunologically active blood cells. These are mainly lymphocytes called T lymphocytes and B lymphocytes. Also there are macrophages, and they either phagocytose these external material or develop immunologically active inflammatory substances that will take care of these external toxins or bacteria that has migrated in the intestinal wall. Let us first just clear the different terms used related to intestinal barrier. The first one that everybody today is aware of is leaky gut. It is a proposed digestive condition where the intestinal lining allows bacteria and toxins into the bloodstream. It is actually not a scientific term. Scientific term is intestinal permeability, which is defined as a functional feature of the intestinal barrier, measurable by analyzing flux rates across the intestinal wall as a whole or across wall components of defined molecules that are largely inert during the process and that can be adequately measured in these settings. And the third part I want to make sure we understand is protein losing enteropathy. It is a condition in which excess loss of proteins occur through the gastrointestinal tract in the lumen due to different etiologies, from ulcers, inflammation of epithelium, lymphatic obstruction, things like that. The thing is, protein losing enteropathy is protein is being lost or oozes out from intestinal wall into the intestinal lumen. While when we talk about leaky gut or intestinal actually permeability, it is for the substances in the intestinal lumen going into the intestinal wall or systemic circulation. To understand the intestinal barrier function, it will be easy if we think about intestinal barrier as a fence or a wall. You want to make sure that you understand the difference between the two, but if we look between two countries, there is a fence or a wall separating the two countries, and nobody is allowed from one country into the other without certain requirements. So if you look this is a wall which is a solid. This is a wall between Israel and Gaza, where nothing can go through at all, no space for anything to go through. Then there are some fence. You can think about this as a US-Mexico border. Even this fence can be torn or damaged, and through which people can go. Or there are sometimes the fence are a little bit porous, where kind of people can go through even though they are not allowed or kind of permitted. But behind this wall or fence, on the other side, are the macrophages or lymphocytes. We call it border security force. They will actually prevent non-allowable people to go further into the country. But we do allow some people to go through. So we allow some people to go through at port of entry. This is actually San Ysidro border between US and Mexico and San Diego. And you can see there are many different areas through which the cars and people can go through. There are different lights on it, different types of lanes for the different type of people or cars. Similarly is the intestinal barrier. Just like this international boundary between two countries, which allows people with visas to enter, but not other people. Similarly, intestinal wall acts as a barrier, which allows certain material that are important or required by our body to go through. Intestine is not completely kind of shutting off anything and everything going through. It allows certain material to go through, and there are different ways by which it can allow material to go through. And these are important because if any of this is affected, it will affect the barrier function. So first way, the intestinal lumen kind of content can go through the intestinal cell or barrier is by lipoidal diffusion. It means that any material that can be dissolved in the fat can go through the fat containing intestinal lining or intestinal cell wall, which is made up of lipopolysaccharide or fat and carbohydrate. So it can allow material to go through. The second way is kind of mediated by the receptors. It means that intestinal cells have a special receptor which attaches to certain protein or sugar in the lumen, and then it is allowed to go through as long as it can attach to the receptor. The similar mechanism exists for certain material secreted in the intestinal cell to go out into the lumen. And these are many times like cytokines or kind of secretion from the intestinal cells responsible for maintaining immunity. There can be secretory IgA and other kind of peptides that can go in the lumen through this receptor, and where it can act or prevent bacteria going through. The other way that intestinal lumen kind of material can go across into the intestinal wall is in between the two cells through this tight junction. We mentioned that there is a tight junction between two cells, but some material like water and some very small ions can go through because they are smaller than the tight gap or tight junction. The last way the material from intestinal lumen can be in the intestinal cell or the wall is endocytosis. Endocytosis is that material, which may be either bacteria or toxin or large like peptides, can be engulfed by the cell wall and then quote swallowed in. And this is way it can be managed here by the immunologic cells or can be transported into the intestinal wall. So these are the different ways the intestinal lining or mechanical barrier will allow certain things to go through. It allows very selective material to go through, and when it cannot allow only selective material go through, then it will be called more like a leaky gut or increased intestinal permeability. So leaky gut is the one on this right hand side. The left hand side is a normal gut or normal intestinal barrier function. You can see there are good bacteria, good mucus layer, very good intestinal lining layer with very tight junctions, preventing anything going in between the cells, and then the blood cells also having normal immunologic function. In leaky gut, any of these could be affected, and that's one is paracellular pathway between the two cells. They may be not as tight, so that there is a space between the two cells. There is a space between two cells through which things can go through, even though they are not supposed to. There may be problem with intestinal mucus layer or bacteria. So to understand intestinal barrier function and to see any defect in that, we'll have to go one by one of these entities. So if we think what might cause increased intestinal permeability, is first one is disruption of microbial kind of milieu or intestinal dysbiosis. There are different type of bacteria or no bacteria or bad bacteria. Second is decreased mucus layer. So this is the mucus layer. It is decreased in density or depth. Also it may be not as thick. The third part is epithelial damage and the disruption of tight junctions. So epithelial damage, intestinal epithelium is somehow damaged, which makes these tight junctions abnormal or opened up. So there is a space between the two cells through which material can go through. And the last one is intestinal immune dysfunction. It means the intestinal wall has macrophages and lymphocytes, and those are not working right. So any of these can cause increased intestinal permeability. So how do we measure intestinal permeability? We want to see that molecules that are size larger than the tight gap junction or tight junction should not pass through the intestinal wall. So if we look in this diagram, that there are two molecules we chose. One is mannitol, which is 6.7 Armstrong unit, and lactulose, which is 9.5 Armstrong unit. 1 Armstrong unit is 10 to power minus 10 meter or 0.1 of nanometer. So it's a very small size. The size of the tight gap junction is, so that's the gap between the two intestinal cells. The size of that gap is around 6 to 7 Armstrong unit. So if you look that way, then mannitol, which is a small molecule, will pass through the intestinal wall or in between these two cells. It will pass through because it is smaller size than the tight junction. While bigger molecule like lactulose, which is yellow here, will not be able to go through unless there is little increase in gap size between the two cells. So what we do is we give two molecules, one is lactulose, another is mannitol in a solution, ask patient to drink, and then whatever is absorbed through the intestine into the blood will be excreted in the urine. So we can collect the urine sample, usually about 2 hours and sometimes up to 5 hours, and then see how much of lactulose and mannitol is excreted in the urine. None of these two substances are found in normal human body. So whatever is in the urine has come from absorbed mannitol or lactulose. Many different molecules have been used in testing intestinal permeability. So some are smaller size than the tight cap junction, so that can be used instead of mannitol, and those are D-xylose, rhamnose, or mannitol. And there are other ones that are larger than the gap junction or size of the tight cap, and those are actually EDTA, lactulose, PEG, which is part of a type of what we call Miralax, DTPA, or other different solutions there have different size molecules. So what is the normal intestinal permeability? It is a ratio of excretion of lactulose to mannitol. Lactulose is normally not absorbed from a normal intestinal epithelium with normal tight junctions. As you can see in this, only 0.35% of orally taken lactulose is absorbed and then excreted in the urine, compared to mannitol, which is smaller size molecule, get excreted up to 12.3%. We then take the ratio of lactulose excretion to mannitol excretion, and then we look the median ratio in healthy person is 0.03. It has a very wide range of 0.0029 to 0.2510. We do not normally use intestinal permeability testing in just a routine clinical practice. It is not much of help or required for regular patient care. However, it has been used in some research settings at the same time also in some cases to detect intestinal epithelial changes where intestinal biopsy do not show any specific abnormality. So these are the cases in some food allergies, celiac disease, Crohn's disease or inflammatory bowel disease, and type 1 diabetes. Also, it can be used as a screening of the high-risk group, such as first degree relative of the patient with celiac disease and Crohn's disease. And they may have an increased intestinal permeability but have not developed disease yet. During the family screening, you may find someone with increased intestinal permeability but may not have developed disease yet. And it has been shown that it can be months or years before they develop the disease that they may have increased intestinal permeability. In one case, patient developed Crohn's disease after 8 years of knowing that they have increased intestinal permeability. So this is still evolving science. Whether to use this as a screening test is very, very early stage, and we do not use it currently. So now we will go ahead and look at what causes increased intestinal permeability. We had looked at this before, and I will come back to these slides multiple times as we check each one of of these mechanisms. The first one is the disruption of microbial milieu or intestinal dysbiosis. So gut microbiota is microorganisms in the intestinal lumen, and this part is the small intestine, this is the large intestine or the colon. Small intestine is villi, while large intestine do not have villi, they just have crypts. The type of bacteria or microorganisms in intestine and the colon differs. Also, the amount of these microorganisms, they increase as you go from small intestine to colon to the end of the colon in the rectum. These bacteria, there are many bacteria, they live in and above just the mucus layer. This is the mucus layer, this is the lining of the colon cells and intestinal cells. There is thick layer of the mucus, above it is the thin layer of the mucus, and just above that and intermixed with this thin layer are some bacteria or microorganisms. There are good bacteria and there are bad bacteria. So these bacteria, good bacteria we call, they compete with this bad bacteria for the nutrition in the intestinal lumen, as well as just sheer number of these bacteria will prevent bad bacteria going further in through the mucus layer into the intestinal lining. Also, these bacteria use the mucus for their nutrition, and they develop what we call outer membrane vesicles, which are taken up by the intestinal cells, and then lymphocytes in the intestinal wall develop some antibodies against these vesicles. It also secretes what we call secretory IgA, which is a protein important for intestinal immunity. These all help in fighting off the invasion from other bad bacteria into the intestinal lining. So these microbiota or microorganisms in the intestinal tract helps the host in many different ways. We just looked. The first is it increases or improves host defense against pathogens. Pathogens are bad bacteria or the microorganisms that cause disease and toxins. Second way, these microbiota help develop and maintain intestinal immune system. And third, that we didn't talk about before, is that it supports digestion of other intestinal lumen content by supply of some enzymatic capacity. And there are some enzymes in this bacteria that may help you digest certain foods that human cannot digest. The most important is the fiber, and the fiber is digested by these bacteria into short chain fatty acids, which are then absorbed in the especially colonic epithelium, where it helps in maintaining as well as providing nutrition to the colon cells. These bacteria also supply micronutrients like vitamin B12 and vitamin K. So these are the ways the gut microbiota may help the host. Any disruption in this microbiota will cause intestinal disease or other disease in the host. So what is this intestinal microbiota? There is the microorganisms residing in the human gastrointestinal tract. We have close to 100 trillion microorganisms in the intestinal tract, and they are almost as many as human cells. So about half of our cell mass is made up of these microbiomes. There are many different types of these microorganisms. We can divide into three types. One is normal bacteria type. These are called good bacteria or helpful bacteria, and they are like lactobacillus, bifidobacterium, other is faecalibacterium. Then there are opportunistic organisms. These are usually not bad organisms unless something else happens where the where the number of the normal microorganisms decrease and number of the opportunistic organisms increase, and then they cause pathogenic problem or some kind of disease in the humans. And the last one, which are definitely bad bacteria or microorganisms that cause disease, and these are Salmonella, Shigella, Clostridia type of bacteria. So what causes intestinal dysbiosis? Dysbiosis is abnormal microorganisms in the gastrointestinal tract, but also the change in the numbers or how they are colonizing the intestinal lumen. The most important cause is use of oral antibiotics and even intravenous antibiotics. It can change the good bacteria, it may kill the good bacteria, so the bad bacteria may overgrow. The second is lack of what we call prebiotics in the diet. So prebiotics are the nutrients that bacteria need to grow. And most important prebiotic is the fiber. The third is excessive FODMAPs, which are type of sugars, mainly in the processed food and very simple sugars, and fat. Excessive amount of this will alter the intestinal bacteria. Next cause is intestinal infection by what we call pathogens. Pathogens are those microorganisms which cause disease in the humans. So these are like viral infections. Common one are right now norovirus, rotavirus, and then bacteria like Salmonella, Shigella, Clostridium infections. All these intestinal infections will alter the microbiota of intestine and cause dysbiosis. Then other way these bacteria may be killed are chemotherapy for cancers and the radiation treatment for cancer. Other one is you may clean out the intestinal tract completely from top to bottom by this cleansing type of procedures people do. Many times these are enemas they take rectally, and then that will remove not just the stool but also the bacteria and the mucus layer. And very frequent use of this type of cleansing practices may lead to removal of many of good bacteria. Other is stress, emotional stress as well as physical stress. Both alters the intestinal microbiota. It has been shown in studies that after very strenuous exercise like marathon running, there is an alteration in intestinal bacteria. And the last one that is just being studied now is how do we acquire this bacteria in the first place in the intestinal tract is through the passage of the baby when the baby is born through the vaginal canal. And during that time, they acquire this bacteria in their intestinal tract. And there may be an abnormal this colonization during this birth time. It has been shown that babies born by C-section have very different intestinal microbiota than those who are born just vaginally. So this can all affect the intestinal kind of microbiota. The next cause of increased intestinal permeability is decrease in the mucus layer. Mucus layer is decreased. Mucus is secreted throughout the gastrointestinal tract from stomach, small intestine, and the colon. The mucus is secreted by goblet cells. Goblet cells are kind of present in the intestinal lining, and they increase in number. Starting from stomach, only 4% of those intestinal lining cells are goblet cells, to slowly increase and go up to 16% in the colon. This is important also that this will create more mucus as we go from stomach to the colon, but also the thickness of the mucus varies in the stomach, small intestine, and the colon. So if we look in the gastrointestinal tract, the mucus changes from stomach all the way to the colon. In the stomach, it is mainly thick mucus layer. In the upper small intestine called duodenum, it is mostly thin layer and very, very few bacteria, because stomach acid kills most of the bacteria, so there are no bacteria usually in the upper small intestine. Then middle small intestine called jejunum, there is little slightly more thicker layer of the mucus with some little bit more bacteria. The lower part of the small intestine called ileum has almost same amount of the mucus, but little bit more thin layer in the top and with little more bacteria. Once we come in the colon, then colon has a big thick layer of the mucus, and on top of that there is thin layer of the mucus. These have many bacteria, and this number of bacteria increases as we go further down towards the rectum, and also the same way the thickness of the mucus layer, which is thick layer, also actually increases in size. So how does this mucus work? Mucus is secreted by goblet cells. Goblet cells are many different types. There are 4 or 5 different types of goblet cells in the intestinal lining. Depends whether it is in small intestine or the colon. They have different types. But main thing to remember is that mucus works by 2 main mechanisms. One is having a mechanical barrier. So you can see that this is in the small intestine. There is a thin layer of the mucus, and it prevents the bacteria from going further down towards the intestinal lining just by having a kind of mucus layer there. In the colon, this layer may actually engulf the colony of microorganisms and prevent it from going further down towards the intestinal lining. The second way this mucus works is it has some chemicals in it. These chemicals, either they are just secreted by goblet cells, or maybe some of them may be secreted by some bacteria. These are antimicrobial peptides and secretory IgA type of antibodies. They prevent bacteria that have migrated from upper layer of the mucus towards the lower layer of the mucus, and when they try to enter into the intestinal epithelium, before that they are caught by these antimicrobial peptides as well as secretory IgA and prevent the bacteria going into the intestinal epithelium. Other than the goblet cells that make the mucus layer is maintained or affected by also the bacteria or microorganisms in the intestinal lumen. Some microorganisms which you call pathogen, like actually cholera or giardia, amoeba, or parasite called trichuris, they induce mucus degradation, so decreases the mucus layer. Some other bacterial and parasitic pathogens, something like listeria, amoeba again, parasites, they also inhibit mucus production by goblet cells. They also kind of regulate the goblet cell function and thereby decrease the mucus layer. There are some good bacteria like lactobacillus. It will stimulate the mucus production. Bifidobacterium mainly restores mucus growth. Lactobacillus may increase the mucus layer thickness, and some other bacteria also kind of increase the goblet cell number and thereby increasing the mucus secretion. Some of the bacteria will use mucus or other nutrients and for their energy production, and they will have metabolites excreted. Most important one are short chain fatty acids, acetate and the butyrate. These short chain fatty acids stimulates mucus expression as well as mucus kind of production and secretion. So bacteria have also large role to play in maintaining the mucus layer. So what are the causes of this decreased mucus layer? Eventually, decreased mucus layer will cause increased intestinal permeability. So first thing is loss of goblet cells, the cells that make the mucus. And these are kind of damaged or dead in case of intestinal infections or any kind of inflammatory bowel disease like ulcerative colitis or Crohn's disease. Other one is dysbiosis, as we saw in a previous slide, that there are some bacteria that are helpful and there are some bacteria that are bad for the mucus layer. So if there is change in these intestinal microorganisms, then it will affect the thickness or the secretion of the mucus layer. The third factor is in the dietary kind of constituents that we consume. If that will affect the mucus layer. Decreased water intake is shown to have decreased thin mucus layer. Other is use of the certain substances in the processed food, mainly the emulsifiers, which are like carboxymethyl cellulose, polysorbate 80, maltodextrin, benzoate, tetrazine. This will make the layer of the mucus thinned out, as they will also kind of dissolve this mucus. Use of pesticides or heavy metals in the agriculture crop is going to affect the mucus layer. And recently it has been shown that nanoparticles from plastics can affect the mucus layer. Just like the microorganisms being flushed out, mucus layer can be flushed out by very frequent cleansing of intestinal tract. Cleansing is done either by enemas or even oral medications that, quote, clean out the intestine, may clean out of bacteria and the mucus layer also. Then there are some very rare congenital kind of metabolic kind of diseases which can affect the secretion of the mucus or type of the mucus. So these are the glycosylation defects. Mucus is a glycoprotein. So if you have any defect in metabolism of these glycoproteins, can cause abnormal mucus layer. And another abnormal mucus we see is very thick or the viscous mucus is in cystic fibrosis. Very thick or viscous mucus also prevents the nutrients also going through this very thick layer of the mucus. The next cause of increased intestinal permeability is epithelial damage and also disruption of tight these junctions. We looked at the mucus layer, we looked at the microbiota. Now we will look at the intestinal epithelial level where the tight junctions are affected. So we have looked at the tight junctions before. This is the small intestinal lining. It has many different cells. These cells are separated by small gap between them, but these gaps are blocked or bridged by these tight gap junctions or tight junctions. If you look at this tight junction, it is made up of proteins called actually clodine and occludine, which are connected to the actin fibers in the cell itself. If there is any damage to this structure, then it will increase intestinal permeability. So in a normal healthy condition, these tight junctions made up of two separate proteins connected internally to the cell, they are very tight and attached to each other. So there is no space between the two cells for anything to go through it. However, in case of any kind of damage to these tight gap junctions or tight junctions, where the cells are separated now by some more space and through which either the microbiota or toxins or other nutrients may go through. So what causes these tight junctions to be dysfunctional? Most important cause will be epithelial damage or damage to the intestinal lining. In that, most common cause is infection and even in that, most common cause is viral gastroenteritis, like actually norovirus or rotavirus infections. Usually, these infections are cured in few days by host immune system. But in some cases, even though the virus or the bacteria is gone, there still remains an intestinal damage as intestine is not healed completely, and that leads to what we call post-infectious enteropathy. The next cause is inflammatory bowel disease like Crohn's disease or ulcerative colitis. There may be intestinal damage from autoimmune disease like celiac disease, which is triggered by the gluten, which makes immune system make autoantibodies against the intestinal lining. Other cause is malnutrition. As you know, intestinal epithelium is has a very rapid turnover. The intestinal cells are continuously made into mature cells from the immature cells, and during that process it needs nutrition. If there is not enough nutrition of either calories or the protein, then it can cause intestinal kind of lining to be affected, and tight junctions may be abnormal or malfunctional. Other is damage to the intestinal cells by radiation or chemotherapy. Again, this is caused mainly in the intestine because of intestinal cells having rapid turnover. And last cause is ischemia or decreased blood supply to the intestinal lining. This may be related to abnormal blood vessels caused by vasculitis, which is in many of the autoimmune disorders, or atherosclerosis affecting the mesenteric or intestinal blood vessels. Another cause for the tight gap kind of junction to be abnormal is psychological stress. The stress can affect the tight junctions by two separate mechanisms. Stress may stimulate hypothalamic pituitary axis, or it can stimulate the sympathetic ganglions or SAM axis, both of which ultimately will lead to eosinophilic cells degranulation or mast cell degranulation, which will lead to release of cytokines or proteoses, which will then affect the tight junctions and increase intestinal permeability. So what can be the result of increased intestinal permeability? There are many diseases thought to be related to increased intestinal permeability. The main in the gastrointestinal tract are celiac disease, Crohn's disease, ulcerative colitis, irritable bowel syndrome, small intestinal bacterial overgrowth. These all may be related as a cause or effect of increased intestinal permeability. This will lead to also some diseases in the liver, which may be alcoholic hepatitis, non-alcoholic fatty liver disease, now called metabolic associated hepatic disorder, cirrhosis of liver, primary biliary cirrhosis, or primary sclerosing cholangitis. In the heart, it may be associated with congestive heart failure. In the lungs, it may be associated with COPD or asthma. In the central nervous system, it may cause depression, schizophrenia, chronic fatigue syndrome, autism, or Alzheimer's disease. It may tip over chronic renal disease into renal failure and has been associated with severe acute pancreatitis or type 1 and type 2 diabetes mellitus. The problem with increased intestinal permeability is once it sets in, then it becomes a cycle where it keeps on going. So because there is change to either intestinal microbiota or change in mucus layer or any other pathologic condition, there is increased intestinal permeability from epithelial barrier failure that leads to microbial translocation that causes mucosal inflammation in the intestine that lead to systemic inflammation, sometimes lead to maldigestion, malabsorption, malnutrition, and continuing this cycle of increased intestinal permeability causing damage. The other cause for intestinal permeability problem is mainly in the diet, and especially the Western style diet, which is rich in fat, particularly saturated fatty acids, which is rich in sugar, particularly in simple processed sugar, something like fructose, and poor in fibers. What happens, it can lead to intestinal kind of permeability problem in two separate ways. One is this diet is energy dense food because it has more fat and more carbohydrate. It changes the energy balance. It means how much energy is derived from which component of the nutrition or the diet. This change leads to change in the microbiota composition, as we had seen earlier. And then this lead to enhancement of energy harvest because this bacteria or abnormal microbiota may have different metabolites which increases the energy load, means more energy is absorbed, which leads to obesity, joint disease, depression. The second arm, the way the Western diet is not helpful, is it causes translocation of bacteria and bacterial products in the intestine because of increased intestinal permeability. This leads to enhanced endotoxin in the portal vein that goes to the liver. That causes low-grade liver inflammation, which lead to non-alcoholic fatty liver disease or metabolic associated liver disease, or MAS, metabolic associated steatohepatitis, leads to insulin resistance and metabolic syndrome. I just want to touch quickly on autism and increased intestinal permeability. It has been postulated, especially in the lay public kind of media, that autism is from this leaky gut. Proposed theory is that leaky gut is secondary to altered gut microbiota, which may have increased Clostridium or Candida species and decreased Lactobacillus or Bifidobacterium. Along with their toxic metabolites, will cause translocation of this microbiota into the intestine, and which will lead to cytokine release, which may include IL-6, TNF alpha, IFN alpha, CCL2, which may cause neurologic inflammation and may be associated with autism. This is just a proposed theory, not actually proven. And now recently, intestinal epithelial dysfunction is also thought to cause increased thrombotic complications in coronavirus disease. We will not go into detail of this, but it has been shown that increased intestinal permeability leads to initiation of this thrombotic state. Another chronic disease that is thought to be associated with increased intestinal permeability is the rheumatoid arthritis. Again, the mechanism is similar. Gut dysbiosis causes increased intestinal permeability that leads to translocation of bacteria in the intestinal epithelium, which releases inflammatory substances, including some cytokines, which may have synovial specificity, leading to arthritis. So it is one of the other chronic diseases thought to be associated with increased intestinal permeability. Diet also plays an important part in maintaining intestinal permeability function. There are many components of the diet. We divide them into macronutrients like protein, carbohydrate, and fat, and micronutrients like vitamins, minerals, and trace elements and antioxidants. In the macronutrient part, protein are the one that are studied most and seem to affect intestinal permeability. One protein, which is gliadin from wheat, rye, and barley, and is the main source of the gluten, tend to increase intestinal permeability and damage the villi, and also increases translocation of bacteria. All these three will increase the systemic inflammatory response and also development of autoimmune disorder. Most of the other protein which is digested into amino acids are mostly helpful in maintaining intestinal permeability. Out of all the amino acids studied, the one that is studied most is the glutamine. Glutamine is important for cellular growth of the intestinal epithelium, and also it tend to improve the tight junctions, and glutamine also helps in making the glutathione complex, which is an antioxidant, and help in getting rid of free radicals. Other amino acids studied are arginine and tryptophan, as well as cysteine, which all directly or indirectly help in maintaining tight junctions and maintain intestinal barrier function. One amino acid, methionine, is thought to kind of decrease the bad effect of the fat that mainly gets converted into lipopolysaccharide, which are thought to be an important for starting or initiating inflammatory response in the systemic circulation. The next is a carbohydrate. The carbohydrate in the form of the glucose or fructose, alcohol or ethanol are also shown to cause the disruption of tight gap junctions. Bile acids, on the other hand, tend to affect multiple sites in intestinal barrier function. Bile acids decrease the mucus layer, affect the villi, and also affect the tight junctions. All of these increases intestinal permeability. Emulsifier, on the other hand, as we have seen before, alter the microbiota. It also alters the mucus layer and thereby affects the tight gap junctions. The next is the fat. Micronutrients in the diet are also very important in maintaining integrity of tight gap junctions. Vitamins A and D help in maintaining microvillus morphology of epithelial cells. They also help in maintaining mucus layer by kind of stimulating mucus secretion, and also help in maintaining the structural integrity of tight junctions. Zinc directly is involved in kind of maintenance of tight junction proteins by being a metalloenzyme. Anthocyanins are very important in maintaining the mucus layer as well as tight junction. Similarly, polyphenols help maintain the structural integrity of tight junctions. So now the main important part. What should we do to support the gut barrier function or maintain intestinal permeability? The first thing is the diet. In the diet, avoid energy-rich Western style diet, which is high in calories but very densely packed calories in small amount of food of either carbohydrate or fat. The second is avoid processed foods because they contain emulsifiers and they also have excessive amount of fat. Avoid high fat foods. Avoid polyols. Polyols are alcohol. They are unabsorbable sugars and sweeteners like xylitol or sorbitol in the candies. that will affect the mucus layer and translocation of bacteria. Avoid FODMAP diet. FODMAP diet is fermentable oligo, di, and monosaccharide and polyols in the diet. This is again mainly use of the processed food or processed sugar in the diet. Avoid certain proteins like actual gluten and casein, especially during and up to 3 weeks after any intestinal infection. During the time of intestinal infection, there is anyway increased intestinal permeability, which may be transient, but during that time, if gluten or casein is consumed, it may pass through the tight junctions secondary to increased intestinal permeability and start an autoimmune disorder or at least some kind of chronic inflammation. So we looked at what to avoid in the diet, but how about increasing certain helpful dietary constituents? So first thing is increase or add prebiotic or fiber. So prebiotics are those that actually provide the nutrition to microorganisms in the intestinal tract, and important out of all these foods is the fiber. Consume 10 to 30 grams of fiber per day, depending on the source of the fiber. Second is increase the glutamine. Glutamine is, we had looked up in the protein part, where glutamine helps in maintaining structural integrity of tight junctions and helps in epithelial cell growth of intestine. Glutamine in the dose of 15 to 30 grams in 3 divided doses may be helpful. Add vitamin A up to 5,000 units per day, vitamin D 800 to 1,000 units per day, and zinc about 50 milligram per day. Also adding the and some of the herbal source of anti-inflammatory substances like the curcumin may help. Other than diet is to add probiotics in the daily use. The probiotics containing lactobacillus, make sure there are more than 10 billion colony forming units, bifidobacterium, streptococcus, saccharomyces. All these are helpful in maintaining the homeostasis of gastrointestinal microbiota. Remember, Baker's yeast, which is a Saccharomyces, it is a nutritional yeast but is not probiotic. VSL number 3 is FDA approved probiotic and used mainly for inflammatory bowel disease patients. Also, fermented products contain some of these probiotics. So, yogurt, buttermilk, Yakult, Activia, kombucha, ginger beer, these also contain probiotics. Daily use of any of these should be considered, but do not use the same supplement all the time. Keep on changing every 3 to 4 months, different types of probiotics, and in the face of any active infection, avoid taking probiotics. Also, do not take probiotics while you are on antibiotics. Other measures that can help improve or maintain gut barrier function are anti-inflammatory substances, mainly actually flavonoids in the dark plants or leaves like berries, dark chocolate, spinach, eggplants. Use of antioxidants, especially polyphenols, which are in vegetables, tea, dark chocolate. The other one is use of short chain fatty acids as a treatment. One, as we mentioned earlier, short chain fatty acids are metabolite of the bacteria in the gut and help in maintaining colonic epithelial health and function. It has been tried in amount of 10 ml twice a day for 1 month and then once a day for 1 to 2 months. Usually these enemas are not available very routinely, but compounding pharmacy may be able to make it. The last one is the chloride channel agonist, like the actual lubiprostone. Lubiprostone is used for constipation management, and it tends to improve the chloride channel that helps in maintaining the tight gap junction function. The most important is avoid stress, any kind of physical or psychological stress, because it has shown to affect the intestinal barrier function. Now, hopefully all of these above will be able to help you in understanding intestinal barrier function and how to maintain the gut barrier function and prevent some of presumed diseases caused by increased intestinal permeability. Thank you for listening to this, and have a gutsy day. Bye-bye.